everyone. It's a pleasure to present the research results on undifferentiated human 8,500 thyroid cancer cell and their sensitivity to Bisphenol A. Study epidemiological and experimental have shown that Bisphenol A increased susceptibility to thyroid disorders, including benign and malignant thyroid nodules. We previously demonstrated that BPA causes cytotoxic effects that are different in controlled thyroid and in hereditary carcinoma, Teixeira and T. These cells exhibited greater resistance to a vapor concentration of 1 mg/ml, equivalent to the specific migration doses recommended as acceptable by the National Health Surveillance Agency and greater doses, suggesting that their mutation could confer resistance to these cells. The objective was to analyze the effects of low bisphenol A dose exposure on anaplastic carcinoma-derived cells, which harbor p53 mutation. We employed the trypan blue exclusion assay to assess cell viability and the cell counting ET assay to measure cytotoxic activity. 16 different concentrations of BPA ranging from 0.1 to 400 AG/ml were tested, exposing the anaplastic carcinoma cells lineage for 24 and 48-hour intervals. The trypan blue assay revealed that anaplastic carcinoma cells were more sensible to BPA cytotoxic effect than control cells, especially at higher than low dose. After a 24-hour exposure to 1 AG/ml, more than 60 control cells and more than 70 of anaplastic carcinoma cells died. Teixeira concentrations of bisphenol A more lethal to anaplastic carcinoma cells. Cell counting kit assay displayed a similar viability pattern in both lineages at lower doses. However, at higher BPA doses, the sensitivity of anaplastic carcinoma cells reemerged. A dose of 12.5 U for male produced much 90 cell anaplastic carcinoma cell death and 45 in 3. We confirmed previous data showing that cells derived from normal human thyroid suffer reduced viability and metabolic activity when exposed to BPA, even at doses considered safe by regulatory agencies. However, contradicting our expectations, anaplastic carcinoma cells were very sensitive to cytotoxic. We conclude that BPA exerts complex and pleiotropic effects on different types of thyroid cells, which may contribute to the difficulty in rating adverse health risks with chronic exposure to low doses. Thank you for your attention.